All right, thank you, everyone. My name is Andrew Ostrenga, or Andy Ostrenga. I'm the chair of the Pharmacy Committee for COG, and I'd like to welcome everyone to the Betsy Poon Pharmacy Continuing Education Series, the 2021 Hot Topics in Pediatric Oncology. If you still need to register to get CE credit, feel free to use this QR code. You can also copy and paste this link. In order to receive your credit, the course code is COG 2021. In order to access your CE credit, once you're registered, you should be able to navigate directly to that in the blue box, or check your email. You'll get an email from CE Synergy. Post-evaluation surveys, please access the post-test evaluation information and overall program evaluation forms immediately after the presentation via the QR code or registration link. If you're a US-based pharmacist, make sure you put your NABP and date of birth to get the form in its entirety. That way it will flow to the CPE monitor. The credit will be awarded per individual sessions. The best abstracts in pediatric oncology will be one session.
Hey,
To access that,
Andy,
yes?
I'm sorry, I don't think we're seeing your slides advance.
Okay. Let me try sharing them again. Do you see them there?
I think they're just not on presenter mode. We see them, they're just not going from one to the next.
Let me try doing screen instead. Now, do you see it?
Yes, thank you.
Great. Well, here's those QR codes. So I apologize for that. I need my coffee. Other details, make sure the flyer in the LMS portal for full details, but in order to get your CE certificate, you must submit all your credits, not just the proof of credits that will go to the CPE monitor through NABP. Do some housekeeping. Make sure you're logged into a good Wi-Fi to make sure you get the whole program. Handouts are available on the CE Synergy portal website. The activity response questions will be presented to measure activity learning. Those will be in the future presentations will be towards the end. Instructions for polling. So you'll be prompted for polling questions through the presentation. A pop-up will appear in the Zoom screen. The options will be given multiple choice questions. You select your answer, and then a couple sample questions will be provided here. So just a few tests for the ARS system if you want to present, list those for the technician. First, what career would you choose if money or talent were no object? Unfortunately, both of those are an issue for me. A professional baseball player, Amazon rainforest guide, interior design, zookeeper, or movie star? Next question, which of the following best accurately describes your interest in board certification credits? BCOP only, BCPPS, BCOP and BCPPS? Neither is important, or otherwise board certification is not an issue. Which of the following online program options do you find most beneficial? A live webinar, recorded webinar, home study, or no preference? And then what range best reflects that you'd be willing to pay for the CE? Less than $20, 21 to 40, 41 to 60, and cost is generally not a factor. So please answer those. We'll do individual speaker welcomes and introductions. I'd really like to thank all those that were involved in this process. Thank you again for the speakers, our best abstracts presenters. The CE Synergy has these disclosures here for ACPE credit. You can see the screen as far as mitigation. Brooke has a small disclosure, and then here are the rest of the committees reviewing, just again, one small one. Again, I'd like to thank both the committee. As you can see, all the members here have done an awesome job helping design and review these different presentations. Our faculty, again, here's their disclosures, and staff's disclosures. Also, really like to thank Jazz Pharmaceuticals as our funding partner. All content for this program has been objectively reviewed by individuals, so there are no conflict of interest there. And CE Synergy and Hot Topics Program Planning Committee are committed to be fair, balanced, and non-biased. As a accrediting provider, CE Synergy attests all the relevant conflict of interest are fully mitigated per standards. Again, thank you for attending today's presentation. I will stop sharing and let Olga do that. And as we're doing this, I'll review this. It looks like interior designer is the leader right now at 35%. That's not what I would have picked. I figured a lot more movie stars would be in there. The leader for the board certification are, looks like BCOP. Most people prefer a live webinar. That's good. And then looks like less than $20. So thank you everyone for that. All right, Olga, looks like we're seeing your general slides. I'll go ahead and introduce Olga. For our first presenter, Olga Militano has been a pharmacist with the Children's Oncology Group for 6 years. She received her Bachelor of Science in Pharmacy degree from Long Island University College of Pharmacy. And after that, she took a job in the pharmaceutical industry and went back to school to obtain her traditional Doctor of Pharmacy, again from Long Island, in 2000. Olga completed a pediatric pharmacy residency at Shands and University of Florida in 2001. She has served several clinical pharmacy specialist positions prior to joining CUG. That includes UConn and Yale, Children's Hospital New York, Presbyterian, and Columbia. Also New York Medical College. Olga has received several industry-sponsored research grants as a co-PI during her time at Columbia and a winner of ASBMT Best Pharmacist Abstract Awards twice. She's a member of several COG committees, including the Chemo Standards Task Force, Pharmacy Steering, and Pharmacy Education. She loves the rapidly changing dynamic of pediatric oncology and stem cell transplant field, enjoys being able to contribute through research endeavors with COG. And with that, Olga, you're now leading the presentation. Olga, you're on mute, I think.
Thank you, Andy. And I apologize for that. Well, thank you so much for tuning in. And it's my pleasure to speak at the pharmacist conference today. And I'm so happy that we were able to honor Betsy's memory with this annual continuing education program. I was hoping to see all of you in person today, but hopefully next year. So my slides are not advancing for some reason. Oh, here you go. Okay, I have no conflicts of interest to disclose, and I will not be discussing off-label uses of drugs. The learning objectives for today's talk is to describe the rationale for chemotherapy standardization task force dosing guidelines, explain the use of asparaginase products in COG trials. Okay, I'm having technical issues and the slides are advancing without me pressing anything. Is there something I'm doing wrong? Okay, sorry about that. Explain the use of asparaginase products and recognize opportunities to optimize clinical trial management. So a lot of you are familiar with COG Pharmacy webpage resources, hopefully. The one thing I wanted to point out, some of the frequently asked questions that I receive is about the biosimilar products used in COG trials. We posted a memo about it a couple of years ago. The infant solid tumor dosing guidelines can also be found on the pharmacy webpage. And the vincristine administration memo that was posted in December of 2020, which I will discuss during today's talk, can also be found on the pharmacy webpage resources. Some of you may be familiar with the busulfan concentration reporting standardization project. Again, there is a memo about it that was posted within the last year as well. So what are the chemotherapy standardization task force infant solid tumor dosing guidelines? This project actually started before my time in COG. I believe it started around 2011, 2012, and took quite a few years to complete. The purpose of this project was really to attempt to standardize dosing and dosing adjustment of chemotherapy across COG trials. The analysis of 29 Children's Oncology Group trial revealed 11 sets of criteria using different definitions for age, weight, and body surface area. The new method that uses body surface area dose banding for infants and children with the standards task force dosing guidelines allows for a smoother transition in chemotherapy dosing as the child grows. And it's more gradual transition from the historic weight-based perspective to the to BSA dosing perspective. Some of you remember when we used to have cutoffs of changing from milligram per kilo to milligram per meter square dosing at, let's say, less than 12 months of age or less than 10 kilos of weight. Sometimes the dosing would jump quite significantly, and the new dosing guidelines actually allow for a more gradual transition and dosing more in lines with how a child grows. The dosing increments consider the deliverable doses based on pharmacy standards for accurate volume measurements and of drug concentrations of commercially available agents. Again, that project was completed in January of 2018. So if there's a change in formulation, then we may need to update those guidelines. And they can be found under the pharmacy webpage. So the future directions for the Chemostatus Task Force is to continue to produce new infant solid tumor dosing tables as protocols being developed. And most of you familiar that the new solid tumor protocols that we're enrolling children less than 3 years of age have the BSA based dosing tables for kids that have body surface area less than 0.6 meters square. And currently we're working on standardizing leukemia and lymphoma dosing guidelines. Again, I don't have a final timeline when this project will be completed, but hopefully in the next couple of years. So moving on to everyone's favorite topic these days is asparaginase. We, at one point this summer, had probably 5 different asparaginase formulations available on the shelves, which is great. We had asparaginase cornucopia, which is quite the opposite of what we used to in prior years with the Erwinaze shortages. So when thinking about the different asparaginase products, and I have to thank Jen Thackeray for the slide because she created it, I'm not that technologically advanced. When you think about asparaginase products, it's easy to break them into the long-acting and the short-acting groups. So the 2 long-acting products are the pegaspargase and Calpeg, both made by Servier Pharmaceuticals, marketed under the brand name listed on the slides. The main difference between the 2 products is the linker that's used to connect the polyethylene glycol molecule to the E. coli asparaginase. And I'll go into that in more detail soon. Now, for the short-acting products, we had at one point 3 available, but it was actually just 2 products because asparaginase Erwinia chrysanthemi that was marketed in the US under the brand name Erwinaze up until June of this year is really the same product as the formulation that was allowed to be used under temporary importation by the FDA, which is chrysanthus-based or Erwinaze with an S. Both products are manufactured by Fortin Biopharma in UK, and they're both Erwinia asparaginase products, whereas the long-acting products are E. coli asparaginase based. And finally, the new kid on the block is a recombinant formulation of Erwinia asparaginase, marketed under the brand name of Riley's that was just approved this summer. And most of you know about it as either RCP or JZP458 before it became commercially available. So of the different asparaginase products, most of you familiar with pegaspargase, it has been the standard care and a first frontline therapy for ALL for several decades now. Kilospargase pegol is currently not available in Canada, Australia, and New Zealand, which are the 3 other countries where we have Children's Oncology Group sites. It is FDA approved for patients less than 22 years of age only. It is not approved for patients greater or equal to 22 years of age. And it has significantly longer shelf life compared to pegaspargase. Crisantaspase, or Erwinaze with an S, which was the product approved for the temporary importation by the FDA from the Portland Biopharma plant in UK, was available up until last week. FDA initially approved temporary importation of one batch, or 8,000 vials, and all of those vials have been distributed as of last week, and it is no longer available through the temporary importation. It is, however, available through the FDA personal importation policy on a case-by-case basis. It does require planning in advance. It may take several weeks to receive, and it does require a significant amount of paperwork. Clinigen will provide Erwinaze with an S through the personal importation policy on a patient-per-patient basis, free of charge for up to a 3-month supply. The reason FDA only approved one batch for the temporary importation is because at the time they were reviewing application for Rylaze, and now with Rylaze commercially available, they don't feel they need to approve any more batches through temporary importation. Asparaginase Erwinia chrysanthemi, that was marketed in US under the brand name of Erwinaze with a Z, was available up until June of this year. As most of you know, Portland Biopharma changed their distributors from Jazz to Clinigen, and Jazz was only allowed to distribute batches of asparaginase Erwinia that were produced by December 31st, 2020, and those batches ran out at the end of June. We're hoping that Erwinaze with a Z will be distributed again globally, hopefully by late of 2021, maybe beginning of 2022. Clinigen is currently waiting on regulatory approval from the FDA to distribute the product. And finally, asparaginase Erwinia chrysanthemi recombinant formulation was FDA approved in US at the end of June of this year. It may be available outside of US through the Managed Access Program by late 2021. So currently, it's only available in US. So I wanted to mention a few words about calaspargase pegol use in the COG trials and the rationale for it. So some of you may know that Servier plans to eventually discontinue pegaspargase. Both calaspargase pegol and pegaspargase are currently manufactured by Servier. Calaspargase pegol, as I've mentioned, does not have safety and efficacy data in adults greater or equal to 22 years of age. The adult trials are planned, but they're not yet recruiting patients. And CalPEG is currently not available outside of US. We have asked Servier several times about the timeline for pegaspargase discontinuation, and we were not given a date or a timeline, but logistically speaking, and this is a personal opinion, I don't think pegaspargase can be discontinued until there is adult data for the use of calaspargase pegol and until they receive FDA approval to use CalPEG in adults. But that's just a personal opinion. So that may take a few years. Most of you are familiar that in the last few months, we have amended our high-risk protocol ALL 1732 to include calaspargase pegol. So amendments 1A and 2 introduced an option to use CalPEG starting in delayed intensification and subsequent phases. There is a reason why we introduced it in delayed intensification or later and not in the earlier phases of therapy. We did not want to introduce CalPEG prior to completion of inotuzumab azithromycin blocks in Arm B, because we wanted to have clear safety data with inotuzumab and clean toxicity profile. The choice between calaspargase pegol or pegaspargase in that trial is left completely up to the institutional preference. It was introduced as an option for sites to use in anticipation that at some point during but possibly before the trial completion, pegaspargase may not be available. So we wanted to introduce the CalPEG option. ALL 1731 protocol, which is a frontline standard risk trial, is currently undergoing similar amendments to introduce CalPEG option either in delayed intensification or later, again, because the blinatumomab blocks precede delayed intensification. Both trial also introduced dose capping for obesity for both calaspargase, PEGL, and pegaspargase, and I will go into a little bit more detail about it on subsequent slides. So just to talk briefly about comparison between pegaspargase and CalPEG. So as I mentioned before, the main difference between the two products is the linker that connects polyethylene glycol moiety to E. coli asparaginase. Pegaspargase uses succinimidyl succinate linker or SS linker, whereas CalPEG uses SC or succinimidyl carbonate linker. That linker is what is responsible for the difference in pharmacokinetics between the two products. So the half-life of pegaspargase is somewhere around 5 to 6 days. In asparaginase naive adults, it could be as long as 7 days. The half-life of CalPEG is 16.1 days, so it's almost 3 times longer. In terms of administration, pegaspargase can be given IV over 1 to 2 hours or IM, whereas CalPEG can be only given IV. Now, the prescribing information states that calaspargase PEGL has to be given IV over 1 hour. However, recently published data from DFCI 11001 trial suggests that 1 to 2 hour infusion is fine. And in fact, the adult trials with calaspargase PEGLs that soon will be recruiting patients are infusing it over 2 hours. So the initial protocol amendments to ALL 1732 had a 1 hour infusion of calaspargase PEGL, but the subsequent amendments will change it to 1 to 2 hours. There is also significant difference in shelf life between the two products, with a shelf life of pegaspargase being 5 months, which is quite short, and almost 3 years of 33 months to CalPEG. That may or may not be significant for large, busy cancer centers where pegaspargase doesn't tend to sit on the shelf very long, but it may be of relevance to adult centers that may not use asparaginase products as often or small institutions. The dosing for patients less than 22 years of age is similar between the two products. It's 2,500 international units per meter square. The main difference is frequency of administration, where pegaspargase is not indicated to be given any more frequently than every 14 days. And calaspargase PEGL has the time between doses has to be at least 21 days. Both products, and I need to update the slides, both products can be stored in a refrigerator or at room temperature up to 48 hours. In terms of asparaginase activity levels, typically, nadir asparaginase activity level is greater or equal to 0.1 unit per ml, and day 14 is considered therapeutic. In this trial, listed at the bottom of the slide that just got published this July, this was DFCI 11001 trial that compared pegaspargase to calaspargase PEGL in patients between 1 and 21 years of age. They saw that 88% of the patients in the CalPEG arm had serum asparaginase activity levels greater or equal to 0.1 units per ml at 25 days post-dose. Now, both arms had serum asparaginase activity levels greater or equal to 0.1 units per ml at day 18. But because of the longer half-life of CalPEG, as you would expect, more patients had elevated or serum asparaginase activity levels at day 25. In the CalPEG group. So getting back to rationale for amending COG protocols and changing asparaginase doses. Last August, the chair of COG ALO committee, Dr. Mignon Lo, reached out to a number of leukemia experts and myself and asked to organize COG asparaginase task force, specifically looking to see, are we using the best and the safest doses of asparaginase in our protocol, and do we need to change anything? About a month later, this call to action was published in Pediatric Blood Cancer, and basically the authors appealed to the pediatric oncology community to determine the safest dose of pegaspargase necessary to achieve steady enzymatic activity and asparagine depletion in patients with high-risk features. So asparaginase task force, COG asparaginase task force, met monthly for about 6 months. We've consulted with young adult and adolescent leukemia experts, Archie Blyer and Wendy Stock. We have spoken to Servier. We have reviewed published literature, both adults and kids, and came up with the following dosing recommendations. The decision was made to follow FDA prescription information dosing for both products. So the age cutoff for both products to use the higher dose, or 2,500 units per meter square, which is the FDA approved dose, was implemented in our protocols. Now, as I've mentioned, pegaspargase pegol does not have safety and efficacy information in patients greater or equal to 22 years of age. So in those patients, you have to use pegaspargase. We also went with the U.S. prescribing information dosing for pegaspargase for patients that were greater or equal to 22 years of age. And you recall our frontline leukemia trials go up to 31 years of age, or 25 in one, 31 in the other. And so for patients greater or equal to 22 years of age, the dosing is 2,000 units per meter square per dose. The caveat to that is that we also allowed, based on review of toxicity, published toxicity data, we allowed for dose capping at 1 vial, or 3,715 units per institutional policy in cases of baseline obesity. When we reviewed published data, we saw that obesity has been linked to increased risk of toxicity for patients greater or equal to 10 years of age. And the obesity was defined per CDC standards, which is greater or equal to 95% BMI for patients less than 20 years of age, or BMI greater or equal to 30 for those that were greater or equal to 20 years of age. We also made recommendations to monitor asparaginase activity levels in patients who received capped or lower dose of pegaspargase or pegaspargase pegol. So getting back to the pegaspargase toxicity risk factors, and I will urge all of you, if you can, to attend the asparaginase breakout session this Thursday, September 30th, from 3 to 5 p.m. Pacific time. There will be hot off the press information presented during the very last talk on links between pegaspargase toxicity, age, obesity, and ethnicity. This data, we just received that data from the statistician, which was based on analysis of ALL 0232 and 0434 trials. So looking at the published literature, this was a pediatric retrospective analysis by Lebovic et al. And 183 patients with pre-B-cell ALL that were between 1 and 21 years of age. And they strictly looked at pegaspargase dose less or equal to 1 vial, or 3,750 units. And what they found is that pegaspargase doses greater than one vial resulted in the increased incidence of thromboembolic events, pancreatitis, and hyperglycemia. The second trial listed by Rausch et al. Was a trial in adults, 100 adult patients greater or equal to 18 years of age, who received pegaspargase during induction. And they received different regimens and different dosing. The one thing that I have listed here on the slide, that 57% of patients received CALGB 9511 dosing, is relevant because CALGB 9511 used the dose of 2,000 units per meter square with dose capping at 1 vial. So that's relevant because more than half patients in this retrospective review were capped at 1 vial. Still, they noticed there was 21% of patients developed a greater or equal to grade 3 hyperbilirubinemia, and the risk factors for hepatotoxicity included BSA greater or equal to 2, a low albumin, and platelets greater than 50,000 at baseline. The data from another adult trial by Wendy Stock supported these findings. They did see significantly increased risk of toxicity and decreased event-free survival in obese patients by various BSA breakpoints going between 30 and 35, BMI breakpoints, especially with BMIs greater than 30. So some of the local considerations, whether to use pegaspargase or phosphatase pegol. One thing I want to mention is for the institutions that are part of the 340B program for the outpatient, the costs of pegaspargase are negligible. It is 5 cents a vial, whereas CalPEG costs around $18,000 to $19,000 per vial. So not only there's a huge difference in purchasing costs for the 340B sites, it's also potentially huge revenue losses, because despite paying 5 cents a vial for the pegaspargase, the 340B sites get reimbursed thousands of dollars per purchased vial. So it's difference in purchasing cost and difference or potential revenue losses when you switch to CalPEG if you're part of the 340B program. However, the longer shelf life of phosphatase pegol, if you recall, I mentioned is shelf life of 33 months versus 5 months with pegaspargase, may be of an advantage for either small and non-340B institution or adult centers that may not use as much asparaginase. A few words about recent changes to the dose modification guidelines in our leukemia trials. You may have noticed that we are now allowing pre-medications with H1 and H2 antagonists plus or minus corticosteroids to decrease the risk of hypersensitivity and the need to switch to asparaginase or linear products. This was based on Katz Brown and Stacy Cooper's paper from Johns Hopkins that showed that universal premedications decreased the number of patients who required the switch to asparaginase or linear products, and also decreased the number of allergic reactions. They had very few silent inactivation, and it represented significant cost savings to the institutions. If you notice, this was published in 2019. To 2019, 2020, especially 2020, we had months and months without availability of Erwinia asparaginase. So a lot of institutions had to get creative how to make sure that patients receive all of their asparaginase therapy. A lot of you are familiar with a paper published by Sumit Gupta that looked at asparaginase administration and event-free survival, and there's a clear link, especially in high-risk patients, between missing asparaginase doses and event-free survival. So some other clinical considerations. A lot of you ask me or email me asking if desensitization is permissible on COG protocol. It is permissible, and we actually adding that language in the dose modification sections. However, we're not going to dictate how you're going to desensitize your patients. There are several publications that talk about desensitization. The choice of the desensitization protocol is up to individual site. The other reminder is that both CalPEG and pegaspargase are E. coli formulation. They should not be used in patients. So CalPEG should not be used in patients with pegaspargase allergies. This is one of the questions I received from a couple of institutions. So if you have a patient allergic to PEG, CalPEG is not a good option. They're both E. coli formulations. Asparaginase Erwinia therapy should begin within 72 hours of pegaspargase or CalPEG reaction, or sooner if possible. Patients with ALL or lymphoblastic lymphoma should receive 6 doses of short-acting asparaginase formulation for every planned dose of long-acting E. coli asparaginase product, either pegaspargase or calped. Again, it's 6 doses. Asparaginase aurinia chrysanthemi recombinant formulation that was recently approved, the summer Rylaze, should not be used in patients who have previous hypersensitivity or anaphylaxis to asparaginase aurinia chrysanthemi, Erwinase with a Z or Erwinase with an S, because again, they all are Erwinia-based products. And this is the CTCAE grading for allergic reaction. If you notice, COG dose modification guidelines for asparaginase often break down the allergy into mild to moderate versus severe. So mild allergy typically considered grade 1 or 2, something that can be either does not require systemic intervention or only oral intervention is indicated. Once you get into the bronchospasm, need for IV intervention or life-threatening consequences, this becomes moderate to severe reaction, often prompting switching to Erwinia asparaginase product. So some of the disease considerations. So for ALL lymphoblastic lymphoma, as you recall, pegaspargase can be given IV or IM, where calped currently can only be given IV. The infusion duration for calpagase pegol can be 1 to 2 hours, and this will be reflected in future amendments. I believe ALL 1732 currently still states 1 hour infusion. The choice of short-acting asparaginase product is up to each institution. We will not dictate what choice, what product you get. It's whatever you can get on the shelf. The dosing for asparaginase aurinia recombinant product should be per US prescribing information. Currently, it's 25 milligrams per meter square IM every 48 hours. However, that may change. Jazz did submit information to the FDA for the Monday, Wednesday, Friday dosing. That is why we intentionally did not put the dosing of Rylaze in our protocol. We anticipated it changing, and we felt that it would take longer to amend our protocol than for Jazz to amend prescribing information. For AML trials, asparaginase, Erwinia chrysanthemi, or chrysanthespase, which are Erwinase with a Z or Erwinase with an S, are preferred products if you're able to obtain them. Pegaspargase and calped should not be used. They are long-acting products. As you recall, in AML, asparaginase is only used in the Capizzi regimen with high-dose cytarabine, and that Capizzi regimen was based on short-acting formulations. We don't know the dose equivalency between 2 doses of short-acting asparaginase Erwinia or E. coli formulation with the long-acting calped or pegaspargase formulation. So if you are not able to obtain an Erwinia asparaginase product or E. coli short-acting E. coli product, then you should omit asparaginase altogether from the Capizzi regimen. We do not recommend using pegaspargase or calped. The recombinant formulation of asparaginase Erwinia Rylaze can be used as a second line. And the reason for that is we don't have data in AML patients. And we posted a memo about that discusses all of this as well as dosing for Rylaze for patients that are treated on AML 1831 and AML 1531, which is the Down syndrome trial that are the 2 currently active AML trials. So moving on to vincristine, and I'm going to try to be brief. Most of you are aware that vincristine prescribing information has been revised to remove IV push in a syringe route of administration. This happened, I believe, last summer. The warning section of the package insert now states, to reduce potential for fatal medication errors due to incorrect route of administration, vincristine sulfate injection should be diluted in a flexible plastic container and prominently labeled as indicated for intravenous use only, fatal if given by other routes. Institute for Safe Medication Practices also issued a safety alert about this in 2020. So last year we made a decision as Jones Oncology Group to have vincristine administered IV slow infusion in a mini bath in all of our trials starting with January 1st, 2022. We were considering exemptions for Australia and New Zealand. However, after consultation with COGQA, we decided not to make any exemptions to this for any of the countries. And if you, in a situation, for instance, where you have an infant and who doesn't have a central line and you want to give it an IV push, then you would file a deviation just like you normally file for a protocol deviation. But we are not making exceptions to this rule. Again, this will affect protocols that are either new protocols using vincristine opening after January 1st, 2022, or amendments to existing protocols. Just a few words about role of pharmacogenomics, and this is something that as a pharmacy committee we have been trying to work on. This is playing more and more of a role in our trials. A lot of you are familiar with Clinical Pharmacogenomics Implementation Consortium, or CPIC, which provides guidelines for implementation of pharmacogenomics in practice. And again, this is now outdated since I submitted the slides. There are 26 guidelines on 90 drugs, and those guidelines include fluorouracil, respiricase, mercaptopurine, thioguanine, and allopurinol. Currently, asparaginase products or vincristine are not part of those guidelines, but that may change in the future. There have been several publications showing association between pancreatitis and carboxypeptidase A2 single nucleotide polymorphisms, also association between SOT2 gene with a specific CC genotype, RS4880 polymorphism and hepatotoxicity, and association with single nucleotide polymorphism at F11, RS203, 6914 gene and thrombosis. We're hoping to look at more of these pharmacogenomic associations in future trials. So there are a number of opportunities for us as pharmacists to optimize clinical trial management. And I want to thank those of you that volunteer your time to be study pharmacists and work with the study committee and with myself to get these protocols written, reviewed. It's a huge community service. Kudos to you. Thank you for volunteering. I learn from you guys every day. So some of you have noticed that probably around 2015, where the pediatric match trial opened, we started transitioning from trials based on diagnosis to trials in which drugs evaluated for activity in biologically defined populations, like the pediatric match, where we have it's an umbrella trial design where there's a screening protocol and multiple sub-protocols that match patient with a specific mutation to a drug that targets that mutation. The other area for improvement is the genomic landscape at relapse in pediatrics. It's largely underexplored, and because most of genomic characterization for childhood cancers have been performed on diagnostic specimens. And just give you one specific example, for instance, you know, a lot of you are familiar with ANBL 1531 trial, which is a neuroblastoma trial that has Arm E that introduced crizotinib for patients with an ALK mutation. Well, in neuroblastoma specifically, the incidence of ALK mutations in the new diagnosed patients are probably 5 to 10%. However, that incidence in patients at relapse could be as high as 30 to 40%. So exploring genomic landscape at relapse is very important. The other thing that I hope to see that we do as a committee in the future is implement pharmacogenomics more and more in our trials. There's definitely a relationship between adverse event and therapy response. We have a well-established relationship in ALL for the TPMT and T15 mutations, and it is incorporated in our frontline trials with mercaptopurine and thiaguanine, but there's definitely room for improvement and introducing more pharmacogenomics in our trial. There's also a role for ontogeny or organ system maturation and how that potentially affects pharmacogenomics. And we also lack pharmacogenomic data, but targeted small molecule inhibitors that undergo extensive hepatic metabolism, which is what a lot of our developmental therapeutic trials are with. We're hoping to establish pharmacogenomics section under the COG pharmacy discipline. And we're hoping in the future to see more COG pharmacy discipline-led trials. So audience response question number 1. In ALL patients with severe pig asparagase allergy, what is the practice at your institution? And I want to warn you, there is no right or wrong answer to this one. It's just a polling of practice standards. Would you attempt desensitization? Would you switch to either Erwinase with a Z or Erwinase with an S? Would you switch to asparaginase or Rainier chrysanthemum recombinant newly approved formulation? Or would you switch to the least expensive short-acting asparaginase or Rainier formulation if more than one formulation is available? And I'll give you a few minutes to answer. I don't know if Metro Connection were able to pull up that poll for people. Okay, thank you. So for now, just answer question number 1, and we'll move to question number 2 in a few minutes. And Angie, let me know when you get the polling results, because I don't think I could see them.
It looks like it's overwhelming switching to asparaginase, Erwinia, or chrysanthemase.
Okay. All right. So question number 2, and I'll leave the poll up. Dose capping of pig asparagase or cholasparagase pegol at 1 vial, which is 3,750 units per dose, in non-obese patients is permitted in COG ALL trials. True or false?
All right, that one looks like 60% false, 40% true.
Well, currently, that would be if you cap a patient at 1 vial and they do not meet obesity definition that I previously described, it would be considered a deviation. And remember that greater than 10% difference between the calculated and administered dose is going to be a major deviation during an audit. The caveat is that you have to do what's in the best interest in your patient. And if you do choose to dose cap a non-obese patient, document the rationale for doing that. So these are the references that I used, and I will now open it to questions. And
Olga,
over time, right?
Yeah, you're a little bit over. So what I did is we had one question, so I copied it to the chat. So this is one long session. So if you have additional questions for Olga, I'll copy it over the chat. And Olga, you should be able to see that, and you can answer it right there in the chat format. So
Okay.
we'll move on to our next. Thank you again, Olga, for that. It was an awesome presentation.